Welcome, my dear students. Now I would like to explain about autoradiography. Imaging of a sample labeled with radioactive isotopes is called as autoradiography. In this method, the sample itself is the source of radiation because of incorporation of radioactive isotopes that emit alpha rays or beta rays or gamma rays. Hence, it is called as autoradiography. Coming to the history, it was first discovered by Marie Curie in 1898. Later, film stripping method and photographic emulsion method were discovered by Roger in 1979. Now we will see the principle of autoradiography. Okay, radioactive isotopes have an inherent ability to emit alpha rays or beta rays or gamma rays until the radioactive decay is completed. Radioactive decay means losing of the energy. Okay, when these radioactive isotopes are incorporated into a sample, either it may be cell or tissue, so that the radioactive isotopes becomes part of the cell or cell organelles or DNA, etc. So that here the cell or tissue becomes source of the radiation. Okay, and then the sample is placed against X-ray film in dark or under safe light so that we can get the image of that particular sample. Okay, once again listen carefully. Radioactive isotopes have inherent ability to emit alpha rays or beta rays or gamma rays, okay, until the radioactive decay is completed. When these radioactive isotopes are incorporated into the sample, they become part of the cell or cell organelle or DNA, etc. Okay, so that the cell or tissue becomes source of the radiation. When this sample is placed against X-ray film in dark or under safe light, so that we can take an image of the particular sample. Now coming to the procedure, in animals, the radioactive isotopes are injected into the bloodstream of the animal so that the isotopes are observed and becomes part of the sample. After some time, the animals are killed by cervical dislocation method and the sample is isolated using various types of method. Okay, the obtained sample is placed directly against the X-ray film for a period of time to allow the radioactive emissions coming from the sample to interact with the emulsion which is present over the X-ray film so that the image of the sample is created. In plants, now we will see the procedure in plants. The roots of the plants are isolated and placed in a medium. That medium consists nutrients as well as radioactive isotopes, so that the radioactive isotopes are absorbed by the plant roots and become part of the plant roots. So then the sample is isolated, same as in the animals. The sample is placed directly against the X-ray film for a period of time. Why? To allow the radioactive emissions from the sample to interact with the emulsion which is present over the X-ray film so that the image of the sample is created. The photoradiography, it is used to find and investigate various properties of the cells or cell organelles or DNA, etc. Okay, it is also used to find the location and amount of particular substance, for example ATP, within a cell, including cell organelles or metabolites, etc. And one more important application of this autoradiography is it is widely used in drug discovery and development. Today most of the drugs are present in the market, so every drug has to be approved by autoradiography. So means here drugs absorption, distribution, metabolism and excretion of those drugs are detected using this autoradiography. And coming to the, it is also used to examine the distribution of drugs in organs or tissues or cells, etc. And one more important application is it is used in elucidation of metabolic pathways. The metabolic pathways, for example glycolysis or Krebs cycle, etc. So that it is widely used in the elucidation means of metabolic pathways. And one more important application is it is used to find the site and performance of target drug. For example, if any drug is given to test it within a animal, so we can check whether it is acting on the same site or not, and we can also check the performance of the drug. Thank you so much.